Hello, everyone, and welcome to the European Frogbit Collaborative webinar series. We have an excellent speaker who's agreed to share his work with us today. Shane Lishawa with Loyola University Chicago will be sharing his work regarding investigating the habitat suitability of European frogbit in the Upper Great Lakes. My name is Nicole Angel. I'm joined by my colleagues Samantha Tank and Autumn McGowan with the Great Lakes Commission. And on behalf of the EFB Collaborative, we want to thank you for participating in today's webinar. So before I get into the meat of the presentation from Shane, I'm just going to go over a brief background on the collaborative. The European Frogbit Collaborative was convened in 2018 when the state of Michigan identified EFB as a priority and got funding from GLRI and along with Central Michigan University to host the collaborative. And once this initial supporting structure was established, an adaptive management framework was developed, and CMU was established as a technical advisory group. And then finally, stakeholders in Michigan were engaged and encouraged to participate. And initially, this work was really Michigan-centric, but as EFB started to spread and become a larger issue, the need for this type of framework really grew. So that's when, in 2021, the Great Lakes Commission was identified to provide neutral backbone support for this framework and was subsequently awarded funds through GLRI in 2022 to do just that and continue to expand the EFB collaborative framework that was put in place by Michigan. So here is a visual depiction of the governance structure for the EFB collaborative. So there's currently 64 members that are part of the collaborative coming from various organizations across the Great Lakes Basin. So the steering committee provides strategic guidance and directs the collaborative's work, as well as speaks on behalf of the collaborative and makes decisions on what exactly to work on. And there are 4 different work groups comprised of subject matter experts that focus on specific aspects of invasive European frogbit. Those include prioritization, management assessment, education outreach, and newly formed the research work group. So the collaborative membership at large is represented by this white box on the screen. And, they really give input on all the products that the collaborative develops. And then, as mentioned prior, the GLC is down there at the bottom, providing that neutral backbone support to the collaborative, including facilitating meetings and just keeping everyone on track to achieve different goals and objectives. So the European Frogbit Collaborative established its regional strategy, which serves to guide the work of it from 2024 to 2026. And the strategy is really founded upon 3 core focus areas, those being regional coordination and collaboration, best management planning, and research and information needs. And this strategy really helps shape what the work groups focus on. And, it's a way to accomplish objectives that the collaborative cares about. And we're about a little over halfway through the strategy. So next, I just wanted to highlight some of the work of the different work groups, starting with the prioritization work group. So the prioritization work group was charged with reviewing information on where EFB is impacting Great Lakes resources, identifying criteria for evaluating sites based on this information, and then developing a system for prioritizing sites for potential management activities. So this work group is currently working on a demo version of a new tool that will help managers prioritize where to both monitor for and manage European frogbit. Moving on to the management assessment work group. The goals of this work group are to develop a tool that managers can use to easily track their EFB monitoring and management actions, and then use that data to help determine best management practices for EFB. This work group recently developed a new field maps application to accomplish these goals called FrogBat, which is the before and after treatment tracker. And a new app SOP was published, and the app is currently in its 2nd year of use, gathering lots of good data. Next, the education outreach work group was charged with informing, developing, publicizing future EFB educational materials at a local and also regional scale. So recently, this group has organized and hosted successful related sessions at conferences, and they're working to develop fact sheets such as those shown on the screen, including the lookalikes fact sheet and a new resource finalized just earlier this month, which is a trifold brochure for educating waterfowl hunters on European frogbit. Next is the research work group, which was charged with coordinating EFB researchers from across the Great Lakes region to build connections with the management community, share research and information updates, and develop shared research priorities. This work group is new as of this past summer, and one of their first objectives involves completing a literature review that documents critical information needs related to EFB impacts on wetland ecosystems. And once that is complete, that will be used to inform a research agenda. And then finally, I wanted to mention the now phased out delimitation work group, which wasn't shown in the original governance structure, just because they've completed all of their objectives as of last year. So this work group is developed a delimitation application where managers can report where they have been, where they've seen EFB, as well as where they've monitored for it, to really help delimit EFB populations across the Great Lakes region. And I just wanted to end my spiel, my introduction spiel, by saying that there's lots of ways to engage with the collaborative community if you're interested. We have a website that hosts archived literature and resources developed, a quarterly newsletter with the latest and greatest EFB information, as well as an amazing collaborative mascot named Froggy, which is shown in the middle of the screen. And if you'd like to learn more about the collaborative or are interested in staying in touch with our different communication networks, we encourage you to visit the website and join our listserv. And then we'll just finish with a few logistical things. So while we strive to minimize technological issues during these webinars, we do apologize for any that might arise. Before we get to our presentation, I'm just going to cover a few logistical items. We've automatically muted all attendees to reduce interruptions. Attendees will remain muted with webcams disabled throughout the webinar. However, we do encourage and will accept written questions from attendees during the webinar. And if you have any questions, please use that Q&A function shown on the screen here. If you have troubles with the Q&A function, I know some folks do, just put your questions in the chat and we'll have Sam move them over to the Q&A. And questions can be submitted anytime during the webinar, but they will be addressed after the presentation has been given. And finally, if you're experiencing any tech difficulties, please use a chat box or email Sam, whose email is shown on the screen. All right. And now it is my great pleasure to introduce our speaker, Shane Lishawa. Shane, you can feel free to pull up your slides as I'm introducing you. So Shane is a senior research associate at Loyola University Chicago School of Environmental Sustainability. His research focuses on investigating the ecology of Great Lakes coastal wetland ecosystems, invasive species impacts, and the ecological response to wetland management, as well as adaptive ecological restoration approaches. Shane works with students and a broad network of collaborators, including academics, ecological managers, community organizations, and tribal nations. And with that, I will pass it over to you, Shane. Thanks for being here. We're seeing it.
Can you see
seeing
me?
it in PowerPoint mode right now, not presentation mode yet. Oh, looks good. Okay, perfect. Take it away.
Thank you so much, Nicole and Great Lakes Commission, EFB Collaborative, for all the work that you're doing. It's greatly appreciated, and for really taking control of the advance of EFB work over the last couple of years. So I just wanted to acknowledge that you all are doing great work, and we do appreciate it. Just a little bit more information about me and my relationship with Frogbit and with this group. So, I initially became aware of European Frogbit by observing it at one of my field sites back in 2010 was the first time that we saw it in the Eastern Upper Peninsula in the St. Mary's River. And before that, and in between then and now, a lot of my work is focused on invasive cattails. And as you'll see from a lot of the data analysis that we've been doing, there's a strong relationship between these two species in Great Lakes coastal wetlands. So it was just kind of a natural, the Frogbit occurred in places where I was visiting for other work initially, and then that eventually led into asking some specific questions about European Frogbit itself, and the research has just kind of spilled forth from there. Okay, so today I'll be talking about investigating the habitat suitability of European Frogbit in the Upper Great Lakes. I encourage questions. We can hopefully have some time at the end to address a lot of those. I have a lot of data in this presentation, and it represents really a body of work from a number of students and a number of papers. So I hope to have time, but I'm actually not sure that how long the presentation is going to end up going. But I'll try to move quickly in order to leave time to answer questions. And I should also not acknowledge my research group, which is a collaborative group. We call ourselves Team Taifa, so you might see that around, and there's a little logo here. So as I was saying, I really feel like I'm presenting, I'm communicating the work of a very huge, group of collaborators, of scientists, of students through this presentation. I'm not going to read everyone's name here, but this is a list of all of the different individuals that have had a significant contribution to the work that I'm going to present today. And this doesn't even include field technicians and summer field workers that have also contributed, which would double or triple the size of this list. So this has been a very big effort amongst many people. In this, you can see that I've got 4 names highlighted and bolded in this on this slide: Louis Yocums, Catherine Wellens, Logan St. John, and Rose Mohammadi. Those are 4 students that Louis, Kate, and Logan all received graduate degrees while doing EFB research, and I'm going to be presenting a lot of their work. And Rose was an undergraduate that did significant work with me. And then these folks, Louis, Kate, Rose, Drew Monks, and Clayton Kingdon, should all really be acknowledged as co-authors because of the size of their contribution to this. I also want to acknowledge right off the top some of the collaborating organizations that contributed to this: the Sault Ste. Marie Tribe of Chippewa Indians, Boise State University, Oregon State University, Michigan DNR for their continued support, Huron Pines, Three Shores SISMA, and University of Michigan and the University of Michigan Biological Station. Also, funders, the Michigan Invasive Species Grant Program provided several iterations of funding that supported this work. Great Lakes Restoration Initiative provided a lot of the funding to support parallel research in cattails that really led to this work. And then I also supported this work through a couple of different internal funding sources from Loyola University Chicago, undergraduate research opportunities program, and then a research support grant. Okay, so getting into the actual meat of of this presentation here today, I outlined roughly what we're going to talk about. Briefly, I'm going to introduce habitat suitability modeling. What is it? What are some of the challenges? We'll talk about some of the specific habitat, like niche studies that my group has done related to European frogbit, and those include observational studies, a facilitation study, and then Great Lakes wide analysis. And then we'll get into Louis Yocum's PhD work, which really dealt with habitat suitability analyses at a bigger scale. And finally, we'll end with some predicting mapping applications, and I'll talk to you about some of the more recent work trying to provide a web interface where we can kind of summarize and create these mapping applications to provide information for managers and for people that are thinking about frogbit on the landscape. Okay, so throughout this presentation, I'll be kind of using the terms habitat suitability modeling and species distribution modeling interchangeably. But essentially, what we're talking about is statistical or spatial modeling that uses biophysical factors to predict species habitat and distribution across the landscape. So using variables that you can measure in the field or remotely in order to predict the likelihood of occurrence and abundance of species. So why would we want to develop these for invasive species? This is probably a pretty obvious question to you all, but the three things that really pop out to me that I want to underline are survey efficiency. So over the last 10 years, there have been SISMA groups around the state of Michigan, and there have been other folks around the region that have been surveying for frogbit, and maps that can highlight likely locations can really increase the efficiency of those surveying efforts. We're limited by resources, and so that's really important. And then in order to actually effectively tackle invasive species in general, early detection and rapid response is crucial. So if we can't find the new locations before they become really well established, it's really a pretty hopeless enterprise to control invasive species. So it's really, really crucial to increase efficiency and for the purpose of early detection and rapid response. And then finally, I see the work that we're doing here in habitat suitability modeling as important for developing workflows that can allow us to be better prepared for the next set of invasive species that we all know are going to arrive in the Great Lakes. So some of the challenges that occur associated with invasive species-specific habitat suitability modeling is realized versus potential niche. So due to the fact that these organisms are spreading throughout the landscape, dispersal factors, proximity to known or to existing populations are maybe more important factors than actually the habitat conditions under which the organism can survive. So it may be that, say, frogbit has a very narrow range of water level conditions, but if it's really spreading slowly and you're looking at right at that kind of bleeding edge of where the frogbit is spreading, the proximity to known populations might be a more important factor. So that has to be considered in habitat suitability modeling in order for it to be accurate. Data availability is another big challenge. In order to train these models, you need to have a lot of occurrence data, abundance data, and that's not always available. And then the third major factor that our work has kind of addressed is shifting niche space. So specifically in the Great Lakes, where with rapidly changing water levels, you might have conditions in one year that have a certain niche space that would be suitable for frogbit, and then the water level goes up 2 meters over, or 1.5 meters over 5 years, and that niche space is going to shift dramatically. So that is another factor that needs to be definitely considered in order to create quality HSMs. Okay, so stepping back just a little bit and thinking about this kind of chronologically and how our lab, our group has gotten to the place that we are in terms of our modeling and our spatial analysis. So one of the first studies that we did was the ecophenology of EFB. This is Kate Welland's master's thesis from Oregon State University. She's advised by Dennis Albert. And she did a study that looked at the relationship between water depth and plant communities and EFB. And this is at Muskong Marsh in the St. Mary's River. In addition to looking at those relationships, she also looked at the phenology of the turion, which is, as you all know, I assume, the vegetative reproductive structure for EFB. So she looked at the germination of turions under different conditions. You can see in this image on the right, she had 6 transects. It's very clear from this picture that transects 4, 5, and 6, this is within an invasive cattail stand. 1, 2, 3 were outside of that invasive cattail stand. She measured a whole suite of variables across all 6 of those transects. I'm just going to pull out 2 of the many findings that she had just to highlight kind of what we learned about frogbit habitat and niche. So Kate found that the water depth tolerance of frogbit appeared and was abundant between 60 and 120 centimeters of water. Beyond that depth, the transects were outside of the cattail, and the water depth was greater, and there was no frog bit in those locations. So this isn't necessarily saying that the water depth alone was limiting the frog bit to below 120 centimeters, but we just did not find it in that deep of water. But this did illustrate that frog bit can tolerate a wide range of water levels. Kind of more salient was the correlation between Typha glauca, this is hybrid cattail, and European frog bit. So the figure on the right here shows percent cover of frog bit and percent cover of cattail, and this is limited to the 3 transects that were within the cattail. There was no European frog bit outside of the cattail stand at all. So in this case, as you walked or boated from the open water into the cattail, you went from basically a bimodal kind of situation where there was 0 frog bit, and then you got inside the cattails and you had very dense frog bit, as you can see in this image on the left. So we had observed this. Kate's work kind of provided some data to support this relationship that we had observed in the field for several years up to this point. Okay, so this then led to the second set of studies based on those observations, the relationships that we were seeing with water level, and then also with specifically with cattails. We wanted to evaluate if this pattern of correlation or relationship occurred at different scales, at the Great Lakes wide scale, and then we did a facilitation study at the local scale as well. So at the local within wetland scale, we wanted to see if how strong this relationship was with cattail. And so in order to test that, we did some aggressive cattail removal work. This image is actually from Alpena. It's not actually from Muskogee, but it illustrates the physical nature of the cattail removal, wherein we harvested the above water cattail biomass in one set of treatments, which is basically like mowing the lawn. It comes back up the following year. And then we also did a second set of treatments where we cut the cattail below the water surface, which is very effective at killing the cattail. And in that, if you have standing water and you cut that connectivity between the atmosphere and the rhizomes, it results in, basically drowns the plant and results in openings in these cattail stands, and those openings persist for multiple years. So we wanted, by cutting below water, we were creating conditions where we didn't actively remove the frog bit. I mean, we did a little bit passively, but we just removed the structure of the cattail and then compared the frog bit color following that treatment. And as expected, but it was actually a little bit more stark than we were really anticipating. We found that, and just to orient you, on the left, this figure is frog bit percent cover, and then this is the average cover by treatment, and this is in the 2 years following our treatments. So we implemented our treatments in 2016, and we tracked frog bit and Typha and a variety of other variables over 2 years following the treatments. And we found that in those below water harvest treatments where there was low Typha cover, which is represented in the right graph here, we had persistent reduced frog bit cover. So this provided evidence to us, pretty strong evidence, that the cattail is responsible in some part for creating conditions that the frog bit tolerates. So this is evidence of facilitation by Typha. Okay, so the second part of this study was evaluating patterns at the regional scale. And we were able to access the Great Lakes Coastal Wetland Monitoring Program spatial or vegetation data from 2011 to 2015 for this study. And excuse me, we broke the spatial the data into 2 different locations. We had established populations, which was Lake Ontario, and we frogbit had been present for long enough in Lake Ontario that we figured that it was undergoing kind of different processes, like the relationship between the frogbit and the environment was likely different in this long-term established population versus in the invading population, which is Lake Erie and up in the Great Lakes. Within all of those two different kind of broken up data sets, we evaluated statistically EFB presence versus vegetation and environmental variables that were available in the Great Lakes Coastal Wetland Monitoring Program data set. Okay, what did we find? So within the established populations, at the site level, so with the data aggregated down to the wetland, we found that EFB presence was predicted by TIFA. That was the strongest predictor variable. In all of these representations of the models, they're listed by the strength of their predictive power. So TIFA was the first variable to predict EFB presence in Lake Ontario at the site level. Counterintuitively, Phragmites had a negative relationship at the site level. And then we also evaluated data at the plot level. So this is the individual meter squared quadrat level. And we found, once again, that TIFA was one of the most important variables predicting EFB occurrence. There was also a relationship with zone, with a negative relationship with meadow zone, a positive relationship with emergent zone, which is not surprising, a negative relationship with standing dead, and then in this case, there was actually a positive plot level correlation with Phragmites. Okay, so in the invading populations, once again, we saw some similar patterns. At the site level, TIFA was a driver. Standing dead, which is actually associated with TIFA to a certain extent. Organic depth and reed canary grass were all positively associated. And at the plot level, we found that TIFA, once again, was the most important driving variable for occurrence, followed by a suite of other variables. Importantly, fetch, which is the distance across the open lake, is a proxy for wave energy and lake wind and wave energy, showed up as a negative relationship with EFB presence, which will come up later. Okay, so in sum, we found that TIFA appears to facilitate European frogbit across a range of spatial scales within established regions and also in invading regions. Okay, so now I'm going to highlight some work that was done at a very small scale that tried to unveil some of the specific, like more detailed analysis of environmental relationships with European frogbit abundance. So this is from Rose Mohammadi's undergraduate work, and we're actually working on getting this published right now. In this site, this is the Alpena Wildlife Management Area, which we assumed that this site was saturated with EFB, so this was a non-invading population. The frogbit had been there for a long time, and it was dispersed across the entire unit that we evaluated. So we were trying to really look at the habitat or the species, the distribution within this site, assuming that it was at some sort of level of equilibrium at this location. And Rose wanted to look at the environmental variables that were most closely associated with EFB at this site. And we measured 23 different environmental variables along with vegetation and EFB abundance data. So really trying to figure out if any of these variables were predictive. And then Rose did a complicated zero inflated beta gammaless regression model, which is basically, in layman's terms, that's like a way to evaluate abundance where you have a lot of zeros in your data. So one of the issues with a lot of species distribution models is they will evaluate occurrence, so yes or no, if it's present or absent, but very uncommonly do they get at this level of abundance. So ecologically, just the presence of a single frogbit plant might not be causing ecological impact, whereas 80% cover frogbit might be a point where you're having like major impacts on the ecosystem. And so we were trying to work out on methods in order to assess not only occurrence but really abundance of the frogbit. And one of the issues with that is statistically you end up with a lot of zeros, which are hard to account for, and that's what this statistical modeling approach was attempting to do. So using model selection approaches, Rose was able to identify the strongest set of environmental variables that were correlating with EFB abundance. And those were TIFA biomass, water turbidity, phosphate, floating vegetation, chloride, and above water detritus had a negative relationship with frogbit abundance. And then all the other variables that I mentioned were positively correlated with abundance. Interestingly, these factors were associated, turbidity, phosphate, floating vegetation, and chloride are all associated with reduced water quality, especially the chemicals there. So we have some evidence to indicate that frogbit is positively related to disturbed waters and urban waters with higher levels of pollutants in them. Okay, so at this point, I've gotten through talking about some of our basic analysis of relationships between habitat and European frogbit. And now we're going to get into the habitat suitability modeling at more of a regional scale. Okay, so again, the challenges for invasive species that I want to highlight is this realized versus potential niche. A spreading population of an invader doesn't necessarily occupy all of its available niche space, and that needs to be incorporated into modeling approaches in order to adequately describe where it's going to be. So for invading species that haven't filled that potential niche, dispersal factors can be as important as habitat suitability. So a solution would be, as I just said, to include spatial, human-mediated, dispersal-related, and biophysical factors in these SDMs. And this brings us to the second chapter of Lewis's dissertation, assessing the environmental and dispersal-related drivers of an invasive aquatic plant, EFB, in Great Lakes coastal wetlands. Specifically, he set out to test SDMs that include dispersal and environmental drivers to not only evaluate occurrence, but once again, get at this abundance value, which we determine, is really important ecologically. The data set the data sets that were used in this analysis included the Great Lakes Coastal Monitoring Program data for the state of Michigan from 2011 to 2021. So this was a broader window of time than the subset that we used in our initial analysis. Also included MISN data and I believe some data that we collected in the field ourselves, the other paper, but at least the Great Lakes Coastal Wetland Monitoring data from 2011 to 2021. Okay, so in these models that Lewis was wanting to test, or Lewis tested, was the occurrence points that also have abundance data, and a set of predictors, which include field measured data, which is vegetation cover, European frogbit cover, water depth that were measured in the field, and then also geospatial data, including from some of our other work, predicted human introduction points, boat launches, and then also this fetch value, which I highlighted from the Monk's paper, was negatively associated with frogbit. And then also spatial random effects for the statistical modeling. Lewis employed a Bayesian hurdle model, which allows for modeling both abundance and presence. I'm not going to go into the depths of the statistics in this presentation. You can ask Lewis about those later. Okay, so what did we find from this work? Specifically related to occurrence. So in this figure here, we have occurrence on the top and each of the different variables, and then their effect size. So how much they're influencing the EFB response outcome. And then the second set is cover values. And where you have overlap of these error bars over this one line, that indicates that there is no effect. So this is basically equivalent to a no measurable effect. We found that occurrence was predicted by these spatial and environmental variables pretty strongly. And there were no meaningful effects from the same predictors on abundance. So that's kind of interesting, but I'm going to then focus on our occurrence data from here on up. Okay, so first, as distance from populations increase, the probability of EFB occurrence decreases. So right here on this graph, this is an effects plot of the logistic relationship between distance to nearest population and EFB occurrence. You can see that as you get farther away, as you would expect, in the spreading population, you have a reduction in EFB occurrence. Spatial effects explain approximately 30% of the variation in EFB abundance. Next, we saw a fetch effect. So as expected, with increasing fetch, you had decreasing EFB occurrence. Third, we have a water depth relationship that is nonlinear, where we found a negative or a positive correlation between 0 and 75 centimeters and a negative correlation between 75 and 200. This kind of highlights that there's an optimal range, water depth range for EFB, roughly between 25 and 125 centimeters. And then finally, once again, cattails, invasive cattails. As cattail cover increases, the probability of EFB occurrence also increases. So the key takeaways are that this dispersal-related proximity variables and ecological drivers, water depth, typha, and fetch, are all important predictors for EFB occurrence. Spatial proximity was the most important factor predicting EFB occurrence across the state of Michigan. And the lack of effects on EFB abundance kind of indicates to us that this is in its early stage of invasion, and it's possible that over time or in more saturated locations, we might see stronger relationships between abundance and some of these variables. Okay, so that kind of addressed some of the first challenges, the realized potential niche. The second set of challenges relate to data availability and this shifting niche space. And when I say data availability, we're thinking about how do you model the likely occurrence across the landscape when you don't have continuous data coverage. So if, say, water depth is a very important variable, as we've kind of seen, or typha cover, you would want to use the data that represent those variables in order to create a map that gives you relative probability of frogbit occurrence. But those data are not available, synthetically across the whole landscape. So in the third chapter of Lewis's work, we looked at remote sensing data to try to fill the biophysical and dispersal related data gaps. And also, importantly, incorporating near real-time data to account for shifting niche space. And in this case, we're talking about water level fluctuation. So this is Joachim's 2024 paper in the Journal of Environmental Management. Active remote sensing data and dispersal processes improve predictions for an invasive aquatic plant during a climatic extreme in Great Lakes coastal wetlands. The study region here was the St. Mary's River, east of the bridge, basically from through the Lishawa Islands, and on the U.S. side of the border. Actually, there was some Canadian side of the border too. So through the St. Mary's River. The three research questions that were addressed in this paper were: can satellite data from active remote sensing platforms serve as proxies for ecological factors that drive EFB expansion? And those active remote sensing, we're talking about LiDAR and radar, so SAR and LiDAR data. And then 2, what is the relative influence of dispersal related versus ecological drivers in the current and potential distribution of EFB? This is a lot of overlap with questions we've asked in other studies. And then 3rd, how does a climate extreme, record high water level in 2019-2020 influence invasive plant habitat suitability? Okay, so again, satellite data to look at water depth and TIFA, because those are key drivers, ecological drivers that we've seen from our other work for EFB. And these images just illustrate essentially the same location in a different marsh, Great Lakes Coastal Marsh, in 2012 versus 2020 when I was actually swimming to collect vegetation data. Secondly, again, what factors, ecological and dispersal, are important? And then finally, does this climate extreme, focusing on water depth, influence the suitability of habitat for frogbit? Okay, so Lewis employed just going to open all of these, a machine learning random forest modeling approach to test these questions. And again, I talked about our study region. The predictors that went into this included radar data and LiDAR data for measuring water depth and relating that to topobathymetry, the NOAA topobathymetry data, the radar data used in order to try to get at the vegetation cover and the cattail cover, and then a suite of multispectral data to also help fill in the gaps on vegetation presence absence. So NDVI, NDWI are 2 different spectral variables that were calculated from the Sentinel satellite imagery and used in this model, or these models. And then again, we use a random forest machine learning approach in order to create several of these models and test which ones were the strongest predictors. Briefly, so what did we find in terms of the relationship between the remote sense data and some of these measured important drivers for EFB occurrence? So right here, I have highlighted that LiDAR effectively was an effective proxy for water depth. And so this graph down here is showing in situ, like field measured water depths versus water depths that were extracted from the LiDAR data and associated with the topobathymetry. And you can see there's a really strong relationship. So LiDAR was effective at being used as a proxy for water depth. And the SAR radar data was somewhat less effective for quantifying cattail cover across the landscape. But it was still significant, has a significant relationship, but it was more of a 0.3 relationship instead of a 0.88 plus relationship with the water depth. To question 2, the results, we found that the model that had all of the predictor variables, including the vegetation, SAR derived, the LiDAR, multispectral imagery, fetch, and distance to boat launch, produced the best quality predictive model. And what you can see here in this image is EFB habitat suitability from high in a yellow hue to low in a purple hue across the study region. Additionally, we ran these models using 2 different water levels that represented the high and low across the study period. So the high being roughly the 2019 water level, or 2019-2020, and the lower water level, which is still actually not that low, but relatively low water level in 2022. And the implications of that were that we were able to then generate probability models of likely occurrence, high probability habitat suitability for frogbit under these 2 different water level scenarios. And this kind of illustrates the importance of incorporating relatively real-time water level data into these predictive maps, or else you're going to really miss the likely extent of frog habitat. And what we were able to drive was that with water level decreasing, there was less available EFB habitat across the landscape. So when you had flooded conditions during the high water period, there was more available highly suitable habitat for European frogbit. Okay, so and I understand that I am a little bit over what my 30 minutes allotted already, but I have a few more minutes just to finish up with the predictive mapping applications section of this. So hopefully that's okay with everybody. So now taking all that together, a lot of information. What have we learned, and how can we apply what we learned about habitat to create useful mapping tools and applications that can be utilized by the public and by agencies and by managers? And a lot of this work has been led by Clayton Kingdon, who has really the chops in the coding sphere, and has been able to work through the kind of complicated, at least to my eye, interface between statistical modeling and creating useful applications that can be used on the web. So what we're really trying to do is take this high quality habitat suitability model that was presented in the Yoakum's 2024 paper and scale it from the St. Mary's River to the Great Lakes scale. This is a model workflow of the different data that I kind of went through largely that are going into the species distribution model. So this includes bathymetry, water level elevation, informed by the NOAA gauges. It includes wetland extent. It includes thresholds for EFB tolerance. And then the model includes boat launches, local dispersal, fetch, and the satellite-derived vegetation cover and water depth variables, all getting pumped through the random forest algorithms to produce the species distribution probability map. Again, our desired extent is Great Lakes wide. And initially, we were trying to work with Google Earth Engine, which is what the GEE stands for here. These large data sets would need to be uploaded to GEE. So everything in green here are data sets that are not available on the Google Earth Engine server. And so we would need to upload them. These are very large data sets. Terabytes of data would be required to upload to that server. It's beyond the capacity or the allowance of Google Earth Engine to utilize or to bring all of the data into that system. So that presented an issue. So how are we going to create these accurate models that are incorporating the satellite imagery, real-time water level estimates into an application that can be used Great Lakes wide? And so Lewis has been working on the STAC, Spatial Temporal Asset Catalog system, as an alternative way to organize our data in order to allow sort of real-time modeling of habitat suitability for frogbit. This is the Radiant Earth Foundation is a nonprofit that is kind of spearheaded this. It's the structure of the stack is hierarchical. So essentially, we've created a catalog that encompasses our entire project, and then within that, there are different levels of data, collection, item, and asset, which goes from large scale to small scale. This just to cut this a little bit short, so the stack system is becoming kind of more standard for geospatial data distribution. There's a growing community of users. This is a really good alternative to using like Google Earth Engine. The advantages of Stack is that you can use disparate data sources to bring into a Stack system. It's not practical for us to store data locally or pay for cloud storage space. So what the Stack does is essentially an analogy that Lewis has been, it's an analogy that Clayton's been using, is the difference between Netflix and Blockbuster. So Netflix, you're streaming video. Blockbuster, you have to walk down the street or drive to town and check out a tape and bring it home. So the Stack system allows more of a Netflix level streaming from the cloud system to utilize your data. So you don't have to house it on site or in a server and process it locally. It would allow for spatial designation. You could draw a circle around your area of interest and stream basically the data and model within the site or within the Stack system in order to create an output that doesn't require the huge data use that it would at the Great Lakes scale. Okay, so some of the major advantages is that the web app could access data via our project API, just kind of highlighting that point. So again, you could create a web app that isn't drawing from all over the internet and from different sources. It could just draw directly from the Stack, which is then taking data from different sources. The bad news is that this is kind of a newer system. It's still being kind of integrated. And some data sets are not yet free, and there are obviously technical challenges bringing this all together. So this is just a quick screenshot of the Stack API for our data set here. And you can see within here is the hierarchical setting. We've got local collection of data with fetch, for instance, would be data that would be housed on a server that we have access to. And then there's remote data as well, like the Sentinel satellite imagery that's also in here, and the NOAA DEM data. So again, this is providing like an interface for data that are collected at the local and at the regional scale or in external servers. And this is just an example of some of the Sentinel imagery that you can actually display in the Stack interface for different regions. So this is would allow us to run a model and generate output at a scale that is relevant and isn't too data intensive. So in summary, the Stack API would allow us to directly draw, a web app could directly draw data from the Stack. Again, this Netflix versus Blockbuster idea. So data and modeling outputs stream from the cloud on the fly instead of downloaded and generated on local computer and server. There's major advantages to that. This should allow for regional SDM modeling with real-time data or data that are updated, like water level data, could be relatively real-time updated to the Stack and then incorporated in models, and satellite imagery as well, so that you could be producing with the same structure. You could be producing, a model for the next growing season as opposed to starting from scratch each time and creating these models on a local server. We also see this as the workflow is adaptable to other invasive species, which I think is really important. That's kind of one of the major take homes here, is that this background data structure should be applicable to the next set of invaders that come in. Okay, I'm now going to summarize the entirety of what we talked about today in a couple of quick slides, and then we can hopefully get to a couple questions here. So, summary of key findings for habitat. Again, we found that TIFA appears to facilitate European frogbit across a range of spatial scales within established regions and invading regions. So if you're looking for European frogbit, look in cattails. It really does seem to be a strong, well established correlation between those two taxa. EFB occupies an optimal water range of 0 to 100, and I should modify that to potentially 25 to 125 centimeters. Factors associated with reduced water quality, phosphate and chloride, are positively correlated with EFB abundance, at least in one site. I think we need to do a little bit more work on that to see if some of these pollution variables are really driving EFB at a regional scale. In terms of habitat modeling, we found that dispersal factors, including distance to EFB populations and boat launches, may be more important than habitat suitability for predicting presence. So because EFB is still spreading, it has not yet saturated the environment, these dispersal factors are really critical to consider. Second, remote sensing data can act as a proxy for important habitat factors, specifically TIFA and water depth, and that's real critical in order to create mapping outputs. And then third, water level drives dramatic shifts in the availability of highly suitable habitats. So at least in the Great Lakes coastal zone, it's very important to consider water level and year-to-year water level differences in order to assess suitable habitat. And then finally, predictive mapping applications. The first take home is dynamic mapping with frequently updated spatial data is necessary to accurately predict EFB habitat. And specifically, I'm talking about Great Lakes water levels in terms of the spatial data. Secondly, large data sets require the use of novel data organization tools, such as Stack for cloud-based data and modeling. And then finally, the workflow generated for EFB could and should be adopted for other emerging species. Okay, so with that, I want to open it up to answer some questions. And I know Clayton is here as well. So if there's any specific technical questions about the stack, I would direct you to him because he's got a more comprehensive understanding of that work.
Awesome. Thank you so much, Shane. That was really interesting. A lot of collaborations, just building and building on one another. So really interesting. Thank you so much for sharing. We will now open it up for questions. As a reminder, put your questions in the Q&A. If you can't put them in the chat, we'll move them over there. And I know we're a little close on time, but if you all can stay on, Shane and collaborators, the questions will be in the recording. So we can stay on and answer a few questions if folks have them. I'm not seeing anything coming yet. I did have one question. I know in the beginning of your presentation, like very beginning, you mentioned that there was a negative relationship between Phragmites and EFB. I'm curious if you could explain why you think you saw that, because we often think of them as co-occurring species. So
yes,
I'm just curious.
Yeah. I should have kind of explained that a little bit more, but so what we found was that was within the Lake Ontario data set from 2011 to 2015. So it was an isolated chunk of data, and that was the only place we found a negative relationship at the plot. No, was it the site level? I think it's at the site level. So my interpretation there is that within Lake Ontario, there were some sites that had Phragmites, and there are some sites that didn't have it. The extent of cattail is so wide. There's so many wetlands that have hybrid cattail in Lake Ontario that I think that was essentially just a residual kind of, maybe perhaps TIFA and Phrag weren't co-occurring at the same locations. I don't think that we should put much weight, I guess, I don't think we should put much weight into that particular variable in that particular model. It wasn't the strongest driver. The TIFA was a stronger correlation. But there might be certain circumstances where you have a marsh that is dominated by cattail and to the exclusion of frag, and you would more likely see frogbit at that location. So hopefully that clarifies. That's always been something that was a little bit of a struggle for us to unpack, and we haven't seen it with any other data analysis, that relationship.
Oh yeah, that helps. Yeah, interesting. I was just curious, so thanks for clarifying. I'm not seeing, oh, we do have one question in the chat or in the Q&A. Curious in the field if you typically found EFB to be free floating or rooted.
Typically free floating. Yeah. I guess that it varies. I mean, so in the cattail stands, where I've done a lot of my work, it's typically free floating. Yeah. But I've also seen, especially in some of the sort of model validation work where we're going out to a whole range of different locations. I did see frogbit, more rooted or more, I guess, like associated with the sediment in shallower water sites. So, but that isn't something that I've been really documenting much in our field work.
Yeah, good question. I'm not seeing anything else coming in. And I know some folks have dropped off probably to other meetings, but I did want to just extend another thank you to Shane and collaborators for being on today. And with that, I will just want to let you know that this webinar is recorded and will be posted on the European Frogbit Collaborative website in the next few days. And if you have any additional follow-up questions, feel free to reach out to us at the Great Lakes Commission or Shane as well. So with that, I will close out the webinar and thank you all so much.